talk about transcription. Transcription is the synthesis of RNA using information in the DNA. There are 3 stages in transcription process, which are initiation, elongation, and termination. During initiation, RNA polymerase binds to the promoter region of DNA. The DNA strands then unwind, allowing RNA polymerase to initiate RNA synthesis at start point on minor strand. During elongation, RNA polymerase moves downstream, continuously elongating the RNA transcript in the 5' to 3' direction. At the same time, the DNA strands rejoin to form a double helix. The final stage is termination. The RNA transcript is released and RNA polymerase detached from DNA, completing the transcription process. Before we move on to transcription, let's briefly look at prokaryotic and eukaryotic cells. Prokaryotes are unicellular cells that do not have a nucleus or membrane-bound organelles, which makes them smaller and simpler. Examples include bacteria and archaea. In contrast, eukaryotes are more complex cells with a nucleus and membrane-bound organelles that have organized cellular functions. Examples of eukaryote include animals, plants, fungi, algae, and protozoa.
Good morning, students! Last week, we already learned about transcription, right? So today, we will find out what you guys don't understand about this topic. So, we will fix it together.
Teacher, I thought transcription occur in eukaryotic cells because they have a nucleus. Since in prokaryotic cells, they do not have a nucleus. So, I think in prokaryotic cells, they do not undergo transcription. Is it true, teacher?
Teacher, transcription happen in cytoplasm for all cells, right? For both prokaryote and eukaryote. I do not see why eukaryotic cell would be different since all the steps are the same anyway.
Teacher, what I understand is RNA polymerase is just one enzyme that does all transcription in all cells. So, I don't think there's any difference between prokaryotic and eukaryotic transcription in terms of the enzyme involved.
What you guys said sounds reasonable and many students think the same way. Now, let's break it down.
Prokaryotic cells do not undergo a transcription process, while eukaryotic cells undergo transcription.
In reality, both prokaryotic and eukaryotic cells carry out transcription as part of protein synthesis. Transcription is very essential because protein synthesis cannot occur without first producing mRNA. Since both prokaryotic and eukaryotic cells need proteins to survive, grow, and function, transcription must occur in both types of cells. Therefore, prokaryotic cells are fully capable of transcription even though they are structurally simpler than eukaryotic cells.
Transcription for both prokaryotic cells and eukaryotic cells happens in cytoplasm.
The actual location of transcription depends on the presence or absence of a nucleus. Prokaryotic cells do not have a nucleus, so their DNA is located directly in the cytoplasm. As a result, transcription in prokaryotic cells occurs in the cytoplasm. On the other hand, eukaryotic cells contain a true nucleus that encloses their DNA. Therefore, transcription in eukaryotic cells takes place inside the nucleus, not in the cytoplasm.
Many students assume that prokaryotic and eukaryotic cells use the same RNA polymerase, but this is not true. Let's see the difference together. For prokaryotes, it only uses one RNA polymerase that transcribes all their types of RNAs. Next, let's go to RNA polymerase in eukaryote. There is 3 types of RNA polymerase that we specialize for specific RNA type, which is RNA polymerase 1, RNA polymerase 2, and RNA polymerase 3. Let's go dive deeper in RNA polymerase. We will look at comparison between prokaryote and eukaryote for RNA polymerase first. In prokaryote, it only uses one RNA polymerase to transcribe all the RNAs. So here are the analogy for prokaryote. Imagine that there is one small workplace and in there all the workers wear the same uniform. It means they do the same thing, which is same function that RNA polymerase in prokaryote. while in eukaryote, we start with polymerase 1 that will produce ribosomal RNA. It will catalyze the synthesis of several kinds of rRNA molecules that are components of the ribosomes, while RNA polymerase 2 will synthesize the messenger RNA. It will catalyze the production of protein, and for RNA polymerase 3, it will make transfer RNA, that it will catalyze the synthesis of tRNA and one of the molecule tRNA. So, for the analogy for eukaryotic cells, imagine that there is big workplace like office. Each worker wear different uniform, which different uniform represent different roles, and each role has a specific task. So, it is same like RNA polymerase in eukaryotic cells.
Let's compare transcription initiation factor between prokaryotes and eukaryotes.
I am RNA polymerase and I want to start transcription, but I don't know where to begin.
Is it promoter?
Without sigma factor, I cannot recognize the promoter. Hi, I am sigma factor. Do you need help? Yes, let's help me bind the promoter.
The sigma factor binds to RNA polymerase, helping it recognize the promoter and start transcription. This sigma factor specifically recognizes the -35 and -10 regions of the promoter. But in eukaryotic cells, things are more complicated. RNA polymerase 2 needs a whole helper team. First, the TATA-binding protein and TAFs attach to the TATA box. Then TF2B joins, followed by RNA polymerase 2 with TF2F. Next, TF2E arrives, and finally, TF2H. With the full team in place, transcription can begin. Now let's compare termination between prokaryotes and eukaryotes. Transcription ends when RNA polymerase reaches a termination sequence. At this point, RNA polymerase stops transcription and releases both the DNA template and the newly formed RNA strand. Transcription doesn't stop at the termination signal. RNA polymerase keeps going briefly until the polyadenylation signal appears. This signal marks where the mRNA is cut. After cleavage, polymerase continues a short distance, and the mRNA is released.
By understanding where transcription occurs and how it differs between prokaryotic and eukaryotic cells, we can avoid common misconceptions and develop a clearer understanding of gene expressions.